Welcome to the learning module for deoxynucleotide triphosphate synthesis. This is Dr. Doug Tefke for Cellular and Molecular Medicine. After completing this learning module, students should be able to describe the overall pathway for the synthesis of deoxynucleotide triphosphates and identify the key enzymes involved. They should be able to explain how ribonucleotide reductase is regulated to produce a balance of all four deoxynucleotide triphosphates, and describe how thymidilate is made from deoxy-UMP, explain the clinical significance of thymidilate synthase and dihydrofolate reductase, and give examples of pharmaceuticals that target both thymidilate synthase and dihydrofolate reductase. Cell division requires synthesis of deoxynucleotide triphosphates. This is accomplished by activating the enzyme ribonucleotide reductase, or riboreductase for short. Riboreductase catalyzes the reduction of nucleotide diphosphates at the 2' position. Deoxyribonucleotide diphosphates are then converted to deoxynucleotide triphosphates by the activity of nucleoside diphosphate kinase. During the reaction catalyzed by ribonucleotide reductase, the enzyme itself becomes oxidized at 2 critical cysteine residues. This oxidation of riboreductase must be reversed in order for more deoxynucleotide phosphates to be produced. The electron donor protein thioredoxin donates 2 electrons to reverse the oxidation of riboreductase. Thioredoxin reductase then reduces the oxidized thioredoxin using electrons donated from NADPH. Thus, if NADPH levels are low in the cell, deoxynucleotide triphosphate and DNA synthesis cannot proceed. The overall activity of ribonucleotide reductase must be regulated to achieve a balance of the 4 deoxynucleotides required for DNA synthesis. This is accomplished with 2 different regulatory sites, one controlling the overall activity of ribonucleotide reductase and another controlling the substrate specificity of the enzyme. When ATP is bound to the activity site, ribonucleotide reductase is switched on. When the activity site is bound by deoxy-ATP, ribonucleotide reductase is switched off. ATP, deoxy-ATP, deoxy-TTP, and deoxy-GTP all regulate the activity of the enzyme via the substrate specificity site, switching the enzyme from utilizing ADP, CDP, UDP, and GDP as substrates. Hydroxyurea is capable of scavenging a free radical on a tyrosine residue in the catalytic subunit of riboreductase. Hydroxyurea is used in cancer chemotherapies and also in some sickle cell anemia therapies. The information in this slide is only presented to illustrate the exquisite regulation of ribonucleotide reductase. But please note the overall effects of ATP and deoxy-ATP on ribonucleotide reductase activity. This is important in understanding how defects in purine degradation produce immunodeficiencies. ATP bound to the activity site switches ribonucleotide reductase on. ADP bound in the substrate specificity site leads to the reduction of pyrimidines first. CDP is reduced to deoxy-CDP. UDP is reduced to deoxy-UDP. This leads to increased levels of deoxy-TTP in the cell. Deoxy-TTP replaces ATP in the substrate specificity site. This induces ribonucleotide reductase to begin reducing GDP to deoxy-GDP and stops the reduction of the pyrimidine nucleotide diphosphates. The resulting buildup of deoxy-GTP eventually displaces deoxy-TTP in the substrate specificity site, causing riboreductase to start reducing ADP to deoxy-ATP, and inhibits the conversion of UDP to deoxy-UDP, and eventually deoxy-TTP. Eventually, deoxy-ATP displaces ATP in the activity site, switching ribonucleotide reductase off, preventing the further reduction of nucleotide diphosphates. Defects in purine degradation result in the accumulation of adenosine, which is then converted by salvage enzymes to deoxy-ATP. The increased levels of deoxy-ATP prevent ribonucleotide reductase from being switched on, since our immune system relies on the rapid activation and maturation of immune cells. Defects in purine degradation that result in the accumulation of deoxy-ATP can result in immunodeficiency. Thymidine is made from deoxy-UMP by thymidilate synthase, using N5-N10 methylene tetrahydrofolate as the methyl donor. N5-N10 methylene tetrahydrofolate is regenerated by two reactions. Dihydrofolate is reduced to tetrahydrofolate by the activity of dihydrofolate reductase. Serine hydroxymethyltransferase then transfers the hydroxymethyl group of serine to tetrahydrofolate to regenerate the N5-N10 methylene tetrahydrofolate necessary for the further synthesis of thymidine. Deoxy-UMP is derived from deoxy-UDP or deoxy-CDP, both products of ribonucleotide reductase. Deoxy-UDP and deoxy-UTP can be hydrolyzed to deoxy-UMP. Deoxy-CDP can be hydrolyzed to deoxy-CMP, which can then be deaminated to yield deoxy-UMP. Cancer cells consume deoxynucleotides very quickly for DNA replication, including deoxy-TTP. This means that drugs designed to inhibit the activity of thymidilate synthase can slow the replication of cancer cells. 5-fluorouracil is an analog of thymine. When it's taken up by cells, it is metabolically converted to 5-fluorodeoxy-UMP, which is an irreversible inhibitor of thymidilate synthase. Structural analogs of folic acid, such as methotrexate and aminopterin, are widely used to control rapidly dividing cells. They work by inhibiting dihydrofolate reductase. While 5-fluorodeoxy-UMP irreversibly inhibits the activity of thymidilate synthase, folate analogs such as methotrexate and aminopterin competitively inhibit dihydrofolate reductase. This reduces the level of tetrahydrofolate available in cells for thymidine synthesis and also for de novo synthesis of purines. Because methotrexate and aminopterin inhibit tetrahydrofolate regeneration in all rapidly dividing cells, they produce the classic side effects of chemotherapy: hair loss, nausea, leukopenia. Trimethoprim inhibits bacterial dihydrofolate reductase and is frequently used in the treatment of urinary tract infections. Also, recall that sulfonamides specifically inhibit folate synthesis in microorganisms and are used to treat infections as well. Here's a good review of deoxynucleotide biosynthesis. Ribonucleotide reductase converts nucleoside diphosphates to deoxynucleotide diphosphates. ADP, GDP, CDP, and UDP are its substrates to produce deoxy-ADP, deoxy-GDP, deoxy-CDP, and deoxy-UDP. Its activity is turned on by high levels of ATP, and it's switched off by high levels of deoxy-ATP. Allosteric regulation at substrate specificity sites controls the reduction of specific nucleotide diphosphates, allowing for a balanced production of all 4 deoxynucleotide triphosphates necessary for DNA replication. Thymidine is only synthesized from deoxy-UMP. deoxy-UMP, is obtained from either deoxy-CDP or deoxy-UDP. This helps ensure that thymidine is only used in DNA. Thymidine synthetase uses N5-N10 methylene tetrahydrofolate as the methyl donor. Tetrahydrofolate is then regenerated using the activity of dihydrofolate reductase. A specific nucleoside monophosphate kinase, TMP kinase, converts deoxy-TMP to deoxy-TDP. Nucleoside diphosphate kinase then converts all deoxynucleotide diphosphates to nucleotide triphosphates for use in DNA synthesis. Several cancer chemotherapies and antimicrobial drugs target steps in this pathway, including hydroxyurea, which inhibits riboreductase, 5-fluorouracil, which is an irreversible inhibitor of thymidilate synthase, and methyltrexate, aminopterin, and trimethylprim, which target dihydrofolate reductase. Here are a few questions to test how well you understand deoxynucleotide biosynthesis.